Excellencies, distinguished representatives of FAO members, FAO's partners, I can see researchers, policymakers, and there are also participants from public sector institutions, the private sector, the civil society, and I'm also glad to see some dear colleagues and friends inside the room. Ladies and gentlemen, good day, everyone. Chikemba is my name. I'm the Deputy Director of the Plant Production and Protection Division here at FAO's headquarters. And it is my distinct honor and pleasure, dear friends and colleagues, to welcome you to this session of the conference on genetic improvement for sustainable crop production systems. Along with my colleague, Noel Anglin, I will moderate this session of 90 minutes. But to start, let me provide some context for this session. Globally, about 80% of all the food we consume are plant-based. This implies, therefore, that the transformation of cropping systems to produce more with less is critical to the multifaceted efforts to transform agri-food systems and hence attain universal food security and nutrition as envisaged in the United Nations Sustainable Development Goals. As the FAO Director General asserted in his opening remarks earlier this morning, the organization, that is FAO, is leading this charge for the transformation of agri-food systems through the implementation of the FAO Strategic Framework 2022-31, and farmers' ready access to quality seeds and planting materials of the most suitable crop varieties is the single most critical contributory factor to this transition to sustainable cropping systems. And increasingly, as we have heard, several instances given today, biotechnologies are used to enhance the efficiency, scale, and precision of the development of progressively superior crop varieties, which are well adapted to the farmer's milieu, are productive, nutritious, and tolerant to myriad biotic and abiotic stresses. So this session of this global conference provides a unique multi-stakeholder platform to review the state of the art in and prospects for the application of crop biotechnologies, including genomics and other omics, recombinant DNA technology, genome editing, synthetic biology, new generation sequencing, bioinformatics, cell biology, etc., in crop improvement. And this 90-minute session, dear friends, is divided into two main parts. There will be presentations by these eminent august panelists, which I will moderate. Then subsequently, there will be a panel discussion that you will then have the opportunity to pose questions to these discussants, and that segment will be moderated by Noel. Then I will come back at the end to provide some closing remarks. So to start, let me introduce the panelists who are not seated in any particular order. If I call your name, you just show by a raise of hands, so you're recognized. So we have Professor Rajeev Varshney. He's the Director of State Agricultural Biotechnology Center, Food Futures Institute, Murdoch University, Australia. But some of you probably know Rajeev a lot more from his prior work on genomics at the CGIAR Center, the International Crop Research Institute for Semi-Arid Tropics, or ICRISAT. Then we have Dr. Daniela Bittencourt. She is a senior researcher at the Brazilian Agricultural Research Corporation that you know more as Embrapa. She works in the Genetic Resources Center, or Senagen, in Brasilia, and has worked on biotechnology and innovation for sustainable agriculture, including on themes such as synthetic biology. Dr. Stella Salvo is the head of breeding partnerships for smallholder farming, Bayer Crop Sciences, United States of America. And Dr. Pooja Mathur is the head of the plant breeding and genetics laboratory of the FAO IAEA Joint Center for Nuclear Techniques in Food and Agriculture at Seibersdorf and Vienna, Austria. Last, but by no means the least, is Dr. Sergio Feingold, who has coordinated the biotechnology program of the Instituto Nacional de Tecnología Agropecuaria, with the acronym INTA, in Argentina since 2014. He leads the Agrobiotechnology Laboratory at INCA in Balcarce, Argentina. And with this, ladies and gentlemen, I will then invite Rajeev to make his presentation. Rajeev, please.
Thank you very much, Chike, and good afternoon, everyone. And let me thank the organizer and especially Chike for setting up this nice session. So can I have my slides, please? So in this session, in my 10 minutes talk, I will like to give a brief overview about some genomics technology and their integration for the crop improvement. I will not go in technical details. In morning, we had really great presentation from Professor Sanwen Huang, who is the world leader in the genomics technology, as well as from other colleagues like Mario Kokomo, Eves or so. So what I will do, that first is, in context of this session, I think the key thing is that we need to integrate biotechnology, different kind of biotechnology, with the crop improvement program, so that we can talk about the sustainable crop production, we can have the improved varieties for disease resistance, pest resistance, nutrition, productivity, etc. And in this direction, I would like to mention that way back in 2005, when I was working here in Europe, in Germany, this was the time when rice genome was decoded. So at that time point, we gave a concept of genomics-assisted breeding, and we shared that our vision that how the genomics technology can be integrated with the crop improvement. And I'm pleased to mention that this has become the integral part of many crop improvement program. At later stage, we also showed that how the genomics technology can be used with the gene bank material or with the breeding population. Together with the phenotyping, we can identify the genes, haplotypes, we can develop the catalogs, and then furthermore, in the context of the breeding, they can be used the different kind of breeding technology, MAVC, marker-assisted backcrossing, marker-assisted recurrent selection, genomic selection, etc. In this context, as Chike mentioned that I was working earlier at ICRISAT. At ICRISAT, we used to work on so-called orphan crops. So what we did that when I was working at ICRISAT, and now when I'm working at Murdoch University in Australia, while working on these organizations and in collaboration with large number of partners around the world, in Asia, Europe, South America, and North America, we have been able to decode the genomes for more than 19 crop species. and also developed the pan-genome for about 8 crop species. These crops are important from the food security perspective in developing countries as well as in the developed world. Next point was that, and here I would like to mention these kind of genomic resources, how they can be used in the crop improvement program from the perspective of sustainable crop production. I would like to give an example of one crop called chickpea, which is an important food legume crops grown in about 14.8 million hectares around the world, more than 12 countries, many developing countries. But one key thing is that chickpea crop is affected by drought. Sometimes you can have more than 50% crop affected by the drought. So what we did at that time point in chickpea, we did not have the genomic resources. So as I said, that in collaboration with large number of partners, especially from China, India, United States, Canada, and Australia, initially we decoded a draft genome way back in 2013. And then after that, we decoded, or we sequenced more than 100 varieties from several countries, which were released in 1948 to 2012. Furthermore, we sequenced more than 429 lines from 45 countries. And we have done a lot of phenotyping for agronomic traits. Also, we decoded the genomes for more than 3,000 chickpea lines, which were coming from all over the world. And then we also identified a lot of superior haplotypes that they can be used in the breeding program. And very recently, we have completed the sequencing of more than 10,225 lines. So as in morning, Sanwan and other people mentioned, now this is very easy to sequence large-scale genome, but the key thing is that how we can make the sense from these resources. So what we did that together with the germplasm from the gene bank breeding population, we used those genomic resources, we identified a genomic region, we call it QTL hotspot for drought tolerance, and we did the fine mapping. We also identified some genes like CATF4V gene, which is associated with drought tolerance. So what I'm trying to say, so together with the genomic resources, germplasm, phenotyping, we reached to the stage where we can say that these are the genes associated with drought tolerance. And after that, we worked with the breeding programs. Here in this slide, you can see 3-4 institutes from Indian Council of Agriculture Research in India. There were some issues earlier about the technical capacity. We worked with the different breeding institutes in India, of Ethiopia, Kenya, Tanzania, etc. So we trained those breeders and we integrated those genomics technology. This paper indicates that how they were able to develop 3 important leading varieties and by introgressing this QTL hotspot. And then in these different countries, you got those different guidelines for release of the varieties. So this is not very easy, straightforward process, but then after introgressing this QTL region, while following these countries' guidelines, for instance, in the case of India, we delivered around 5 drought-tolerant varieties, which demonstrated 12 to 25% yield advantage. And the similar kind of thing as a part of the Gates Foundation project from the Tropical Legumes project, we delivered those kind of varieties in Ethiopia, Kenya, Tanzania. And in fact, not only for drought, but even for many other diseases like fusarium wilt, X-coccata blight. So a lot of varieties have come through those genomic innovation and more than integration of those genomics in the breeding program. Even in one of those FAO documents, they also documented some of these success stories that how the genomics were able to deliver the superior varieties for smallholder farmers. And this is not only that one way that we always, generally what we do that we take the research from developed world to the developing countries. Here we have shown the different ways. Like in Australia now when I'm working there, we are having chickpea, also another important crop. What we have done that recently we have developed a pan-genome for 15 Australian chickpea varieties. And when we did the genome analysis with the candidate genes for the QTL hotspot region, we identified that several Australian varieties does not have that QTL hotspot. So what we are doing that right now, we are introgressing that QTL hotspot in Australian varieties with an objective to enhance the yield under the rain-fed conditions. So this is the way that we are working on those aspects. And now, as we discussed in morning and at later stage in afternoon session, another thing what we are doing now in Australia, we are integrating AI-based technology to enhance the genomic prediction and integrate that in the crop improvement program. So this is the new technology that AI-based approaches, they can be really helpful. Furthermore, nowadays we are talking about telomere to telomere genome assemblies. In many crops, people are working on those aspects. And inspired by this kind of work, together with Chinese Academy of Agriculture Sciences, we worked with the Institute of Crop Science, Professor Shuyung Zhang, and then we have done that genome sequencing of about 17 wheat crop varieties coming from China. And moreover, now we are working that kind of work in Australia. So we have developed recently the genomes for about 15 Australian chickpea varieties. We are having another project where we are working with many colleagues, including Roberto Tobrosa, so that we are decoding the genomes of 100 wheat varieties to develop the pen genome. And we are using this pen genome in Australia now for improving the nitrogen use efficiency in the case of wheat, as well as for the heat tolerance. So what I wanted to mention that these kind of genomic resources can be used for the trait improvement, and similar kind of work we are doing nowadays in horticulture crop. And I know that my colleague, Professor Pete Prentice, he's here, and he's leading several projects. We are working together. He will be demonstrating some of those examples that how the genomics technology we are using in many horticulture crops. Now, Chike also mentioned that we can mention briefly that what are the new technology coming in this particular space. I think that, as I mentioned earlier, we talked about genomics-assisted breeding. Now we think that this is the time that during last 15 or 17 years, GAV has delivered a lot. Now this is the time for GAV 2.0. And in this context, you are having that you can use those genomics technology. We do not need to talk about the beneficial alleles. You can also find the deleterious allele, but the use of the genome editing technology, you can purge those alleles. You can accumulate the superior alleles, and there are different kind of approaches, and this is the time now that to use this technology. Furthermore, and I know that in this session we will have some presentation on the genome editing and many other technology, but one thing that microbiome engineering is also very key biotech approach which can be used in the crop improvement. In the area of the genome sequencing, now we are moving towards the spatial omics, where you can sequence not only the DNA at the whole genome or plant level, but even at the single cell level. And this kind of thing, they can identify the genes and the network so that we can edit, we can use them in the crop improvement program. In context of the breeding, that once you get those kind of genomic haplotypes, catalogs, etc., you can use the different kind of approaches, haplotype-based breeding. You can use the AI-based approaches for the genomic prediction, optimal contribution selection, genome editing. So my feeling is this is the time now that we can go for the fast-forward breeding approaches and powered by the genomics technology. So in summary, what I wanted to demonstrate that, well, genomics-assisted breeding has already delivered many improved varieties. They are grown by smallholder farmers in developing countries. I showed example of chickpea, but we have done similar kind of work in many other legume crops. Now we are working on developing the haplotype catalogs across a number of crops in Australia. Genomics, we are moving towards T2T genome, spatial genomics, and as I said, that now this is the time for fast-forward breeding approaches. One thing I would like to mention that I think this is really important, that we need to train the next generation of scientists, and we need to have the international multidisciplinary collaboration so that we can effectively integrate the crop improvement program with genomics. And not only that, we need to think about the integration with agronomy and many other social science, that this is the way that we can really work on those aspects. With these words, I would like to thank my colleagues and collaborators from Australia, many international partners, and many colleagues from my own lab. With these things, thank you very much for your attention.
Thank you very much. Thank you very much, Rajeev. And please note your questions and comments. We'll deal with all those at the very end of all the talks.
Thank you. Good afternoon, everybody. I'm very pleased to be here and hear these panel members talk about their work in the biotechnology that they've used to make changes on yield and lead to more sustainable crop production. I've also worked in biotechnology for quite a while, and I was very interested to hear about punishment of potatoes. I think that's a new angle that I hadn't thought about in biotechnology. So we do have some minutes to ask some questions of our esteemed panel members, and I also want to thank Chike for inviting me to moderate this. But first, I thought we'd open it up to you all to see if you have any questions for the panel members before we begin. Yes, sir, you were very fast. Why don't you go ahead?
Thank you very much. I think this is a great session. I was still sleeping because I just coming from Canada directly here, but it is a wonderful session that now I don't have any sleep anymore. But I have a quick question to Dr. Feingold about the potato, actually. So potato is a clonally propagated crop. So I'm just wondering, for example, when we do gene editing, and and then what's the best way to remove the Cas9? Because if you are doing clonally propagated things, and then probably the Cas9 will be still there, and then we cannot consider as a GE rather than other GE.
Thank you. I'm sorry, I didn't present the technology deeper enough. We're using protoplast transfection with ribonucleotide proteins. So the GE machinery will get into the protoplast and will change the sequence of that particular cell and then degrade it. I mean, it degrades. There's no expression, there's no DNA in the system. So it's a protein plus an RNA.
Yeah, wonderful. I think that this is great. But now the following question will be, so what's the efficiency of that? Frequency or efficiency?
I'm sorry, it was in the presentation as well. It's very, very high. We use polyethylene glycol at 25% and 40%, and the efficiency it was above 60%.
Beautiful.
for one copy. Mm-hmm. But if you use less, you get more chance to get 4 copies. I don't know. Don't ask me to explain that. But since if you want to knock out the gene, you need all 4 copies.
Yeah.
Knock that out. Yeah. But it's not a problem. We screen 100 plants, and we usually get what we are looking for.
Okay, thank you so much.
You're welcome.
Thank you for your question. Now I'm going to turn to a question to ask all of the panel members, and starting down at the end with Stella and coming on down. In your opinion, what biotechnology advancement, past, current, or future one, has been the most revolutionary in terms of sustainable crop production, and very briefly, why? Stella, would you like to start?
Yes, thank you. I actually did my PhD in tissue culture, somatic embryogenesis. Those of you that are working on biotechnology must know that not all crop species are amenable to the tissue culture environment. And so I think some of the advancements are actually what genes are known that can help induce somatic embryogenesis. That is a critical part of the pipeline. And so we heard about a lot of technologies here today, but one thing to rest assured is that this is very difficult. The work is still difficult. It's not a silver bullet. It does take time. As we apply these technologies to other crop systems, we need to get there quickly. And so the more that we're able to share what we know and what we know works, I think the better off the community is. Thanks.
Daniela?
Thank you. Well, I believe that gene editing actually opened a new door for the development of sustainable crops, mostly because we can actually genetic engineering those crops in a specific and target way without actually considering those crops as a genetic modified organism. So this can open several other applications for different characteristics in the plant, increasing the yield of production, increasing resistance to disease. But for future perspectives, well, I have to think that synthetic biology actually will open other doors for us. I think we are just at the beginning. I have to remember that synthetic biology applied for crops actually are just mostly in the labs. We are still developing the tools and the concepts of it. So I still hope that this will actually change the future of agriculture. Thanks.
Thank you. Rajeev?
Thank you very much for this point. So I think it's difficult to pinpoint one specific biotechnology advancement. But in my opinion, for the technology to be called revolutionary, they need to be accessible in those regions that need them the most. So if I have to pick one, I will say that probably decoding the genomes is one of those key technologies. So for instance, unless you have information about the gene, you cannot go to the other technology. So I will suggest that probably the capability to decode the genome is one of the key advancement in the recent years. And once you get these information, you can get the insights needed to develop the crop varieties, either through genomics assisted breeding, GM, or genome editing. And these kind of technology, they can ensure. But one thing is that when we are doing this thing, they should need to be open and accessible to all, so that we can rapidly improve the breeding pipelines in national agriculture research systems in developing world. Thank you.
Thank you. Pusha?
I agree with Rajeev that it's very hard to pinpoint at one technology. But if I have to pick my favorite, I would go with the incremental changes the world is seeing with tissue culture technologies, whether it is the knowledge of baby boom, bushel, or gift, that makes it amenable to have transformation system and range of crops, which is very, very critical when it comes to, precision breeding, because even if the next generation sequencing methods, what Rajeev mentioned, gives us that opportunity to go at that large scale. However, how do we get into the cells, the system, and make the machinery work? It's going to be very, very important. So I would say that continuous improvement in developing those enabling tools and technologies, including single-cell sequencing methodologies, are going to be very, very important moving forward.
Thank you. Thank you. Sergio?
Thank you. I think I will go for the future biotechnology. And one of the really new technology that is arising that has not been applied to agriculture or sustainable crop production is RNA interference. And it's been demonstrated that it has the power to replace agrochemicals 100%. Also, it's an incredible tool to do some functional genetics, something which is related to what Rajeev is saying about knowing the genes and their functions. And the cost and the feasibility to develop double-stranded RNA massively is dropping down incredibly. So I think I would go for RNA interference. Thank you.
Thank you. This next question is for Rajeev and Pusha. How do you see that genomics and biotechnologies can be made more accessible to breeding programs with limited resources to be able to help them fast-track the advancement and production of more resilient crops? And what do you see as the key bottlenecks to integrate biotechnology into breeding programs?
Thank you, Noel. So if I need to start, so I will suggest, or I will talk more about the genomics research. So in my opinion, as I mentioned, all genomics research should be available in public domain through easy-to-use database and open source software. In terms of the technical challenges, I feel two key bottlenecks. One is when we are talking about NARS program using these technology, then the cost. So we need to bring the cost down. And many of us have worked in past in this direction in Global South. Second is technical capacity to integrate those genomics and crop improvement program to develop more resilient crops. And as I said, that even when I was working with ICRISAT, with the CGR Research Institute, national programs, Gates Foundation, we were leading a tropical legume project in 13 countries in Africa, 2 countries in Asia. So at another thing, macro level, implementing south-to-south partnership is very important, as they can enable the breeding institute that are facing the same constraints to work together and overcome them. So in my opinion, I'm sure that everyone in this room is very aware of the positive impact that well thought out south-to-south cooperation initiatives can have. And if we can have these things, we can make the best use of those technology and address the bottleneck. Thank you.
Pooja, any follow-up to that?
In terms of bottlenecks, Noel, I would say the approaches integrating biotechnology into breeding pipelines have been quite fragmented in a lot of cases, and the molecular tools are often applied as standalone. So it's important that, while we have trained personnel in biotechnology, molecular breeding, or genomics-assisted breeding, or for that matter, even new biotechnologies that we are all talking about in terms of precision breeding, it's important that as solution, we have the capacity development programs that bring in interdisciplinary sciences together. It's not just one technology against other, but it's a toolkit that we need to look at, and making sure that we co-design these biotech solutions together with the breeders, keeping in context the need and the demand of the breeding programs, and designing interventions that fit into the breeding timelines and preferences.
Great, thank you. Next question for Stella. What do you see is the role of public perception play in the adoption of biotechnology and the production of more sustainable crops?
Thanks for the question. Being private sector here, largely the leaders that grew the company were the leaders that grew biotechnology, and we have seen the impact of public perception play a huge role in that. As many of you know, it hasn't been easy. There's backlash for sure, and even more so now, I think the democratization of the innovation, seeing public sector take on the innovation, even without private sector, is critical. That's why I enjoy my job, because that's particularly the intersection that we try to emphasize. There's so many different applications of the technology that can be applied across the board, and it's a shame if public perception continues to drag it down. And so keep sharing those great examples. Find areas to partner to accelerate the innovation. Again, it's good for farmers to have competition and options, so let's give them options. So thanks very much for the question.
Thank you. Certainly with things like transgenic and genome editing, there is a lot of perception to manage, but there's also regulatory policies. And this next question is for Sergio. What do you see as the regulatory status of some of these techniques that you've been working on, and how do they play out in different countries?
Thank you. Maybe it will help if we get help from the IT people and show one slide behind my last slide. If you go to my presentation, I mean, there's a lot of going on now on the regulatory aspects of genome editing. Argentina has pioneered in the legislation based on the definition of GMO by the Cartagena Protocol. And the definition of GMO of the Cartagena Protocol is that changes at the DNA level by means of biotechnology. I mean, it says new combinations of sequences by means of biotechnology. So the policymakers of Argentina were focused on the new combination of DNA sequences. If you change one base, or if you knock down a gene, or if you change one allele for other, it's not new. It was pre-existent in nature. So based on that, in Argentina and in all the countries that are in green, genome edited products are taken as conventional products. So one thing we don't want to do is creating a third category, because we don't need more barriers, between countries. And Europe, that had a very strong position against genome editing, that was considering the same as transgenics, with all these high barrier to overcome, and the cumbersome of the deregulation, now is thinking the same, the issue again. So one thing that UK was, or England and some other of the countries of UK were taking advantage of after the Brexit, was to embrace the idea of genome editing can be used to deliver answers to some of the problems they have with, or the programs they have with the farm to fork deal, or the Green Deal within Europe. So the thing is, all the green countries that are there consider genome editing products the same as conventional breeding products. There's no, with this view, either you have a transgenic product or you have a non-transgenic product. There's no need for a third position, genome editing, how much, how many bases were changed. So we don't need that problem now in the world.
Thank you, Sergio.
I just would like to make a brief clarification. In the case of Mexico, it's not prohibited. It's in process of discussion, as happened in many other countries.
Happy to know that. We can talk later.
So I hope you can change the color. Thank you.
I was not the one that made the drawing, but I'm happy to know that. Thank you very much.
Well, in the interest of time, I know we're starting to run towards the end. I'd like to invite Chike back to come and say a few thoughts and wrap up the session.
Okay. Thank you very much, Noel. This last interaction between the gentleman from Mexico and Sergio, typifies the kind of moving goalposts that we are dealing with in terms of the regulatory, frameworks that govern biotechnologies. And I would think also that this is probably FAO's work cut out for it. The panelists in their presentations, in the questions that they have fielded, they have been quite eloquent, and any attempt by me to begin to summarize the several interventions and illuminating presentations that they have made will be a disservice. So let me just then say that I would imagine that the participants are now asking after the conference, what next? We have had this session, so what? You know, what value added? What I will say is, if we cast our minds back to the opening remarks of the Director General in the morning, he wasn't speculative, he wasn't anything. He was quite assertive as to the role of biotechnologies as one of the accelerators with which agri-food systems will be transformed. And so what we have done is to begin that conversation for the gathering of evidence base that could shape the kind of work that FAO would do in this area, leveraging crop biotechnologies to transform agri-food systems. And some of the examples that we have heard today span from genomics through genetic modification, genome editing, synthetic biology, mutation induction enabled by biotechnology. So we could have a conference on any one of these themes. So what I would ask for, dear friends, ladies and gentlemen, is to stay engaged and to watch this space. FAO will certainly be convening, specific discussions or work programs to enable us generate that evidence base to support our members as they make use of what are evidently powerful technologies in transforming cropping systems. And there is a sense of urgency. If we are wondering why now, you may have also teased out from these several presentations that climate change seems to be reoccurring in virtually all the presentations in one way or the other. That in this bid to produce more with less, our efforts are confounded by climate change. Something that we have also heard all through is the aspect of nutrition. People care about the quality of food that they eat. And let me draw your attention to the fact that there are dedicated sessions during this conference that will be looking at climate change, will be looking at nutrition. There are also other things that we have heard here include different aspects of partnerships, North-South partnerships, South-South partnership, public-private sector partnerships, indicating that no one can operate as an island. Policy is important. We scientists, we tend to be comfortable in our laboratories. We refuse to engage with politicians because we don't trust them. We refuse to engage with communicators because they are snake oil merchants. But in the end, the politicians and the communicators, they shape public opinion, and perception matters a lot. So these are things that we need to pay attention to. So having said this, I thank you all for your forbearance. We went over time, but you kept faith with us and stayed this long. I thank the panelists on your behalf. If we may put together a round of applause for them. And we have been able to hear ourselves because of the audiovisual engineers who we don't even see, but they're working behind the scenes. Of course, what the young people, the young professionals we call the messengers, are also very implemental to our having been able to have this session. A very short session, but I think we can continue the conversations in the course of these 3 days. There is going to be a social segment now. Please engage and converse. FAO is all ears, wanting to hear at the topmost leadership how we can use biotechnologies to transform agri-food systems. And I thank you all very much.